All right, good evening, everybody. We're going to talk about osmosis and diffusion tonight. The reason we're talking about osmosis and diffusion is that we're in the cell unit, and cells have to do something that's called maintaining homeostasis. This is a new word for most of you. If you break down the word homeo and stasis, homeo means same and stasis means same. And so it's the idea that cells need to keep a constant internal environment, that the environment stays the same all the time. Examples of ways in which the environment needs to stay the same: cells need the temperature to always be the same. So your body temperature is supposed to stay at 98.6 degrees. And if it changes, that means something's wrong. Either you have a fever or you're hypothermic, but the temperature is always supposed to be the same. That's maintaining homeostasis. If you don't have enough water, your body triggers a sense of thirst so that your water levels will stay the same so that you'll drink. If you haven't eaten in a while, your body triggers a sense of hunger so that your food levels, your sugar levels maintain or stay the same. And that's all called maintaining homeostasis. Cells need to maintain homeostasis in order to work the most efficiently. And they do all kinds of things to try to maintain homeostasis. So part of those things that they do has to do with osmosis and diffusion. So in order for us to talk about osmosis and diffusion, we're going to talk about a few things you should have learned in junior high science. Just a quick review. So for example, atoms and molecules always in motion. They're constantly moving about. Gases move about the most freely. The molecules or atoms of solids move about a fixed point, but they're still moving. And that energy of motion is called, see if you remember, not potential energy, but you guessed it, kinetic energy. Kinetic energy is the energy of motion, and it is the energy that allows particles to diffuse from an area of high concentration to an area of low concentration. So in the diagram, you see all the little yellow dots moving from one side to the next. They're moving from an area of high concentration to a low concentration. That's diffusion. So here's a question for you. Do molecules diffuse faster in warm or cold temperatures? And I think lots of you probably know the answer. The answer is warm. They diffuse faster if the water or whatever, the temperature is warmer. And it turns out, interestingly enough, that's all that temperature is. It's just a measure of how fast molecules are moving. I'm wondering if I can move there. It's nothing more than a measure of how fast molecules are moving. So when you sense that something is warm, it's because molecules are hitting your face or your hands quickly. And if you sense that something is cold, you're sensing less motion. That's all temperature is, how fast molecules are moving. All right, so, oops, that didn't work. There we go. So what determines what can diffuse? Well, diffusion across membranes is determined largely by the size and the chemical charge of the molecule. Big molecules can't diffuse across a cell membrane, but small molecules can. Things that you need to know for this unit: you need to understand oxygen, glucose, carbon dioxide, and water. They all can diffuse across a cell membrane. They're fairly small in size. Proteins, on the other hand, are enormous, ginormous molecules, like the biggest molecules ever found in the universe. They can't diffuse across our cell membranes. They have to be transported across our cell membranes, which is a whole nother set of notes. We'll learn about that later. All right, so what's osmosis? Osmosis is exactly the same thing as diffusion, only when water is moving from an area of high concentration to an area of low concentration. It occurs across a semi-permeable membrane. Semi-permeable simply means it's a little bit permeable. That means that some things can pass through it and some things cannot. So again, diffusion of water from an area of high concentration to an area of low concentration. So then the question becomes, what does water concentration actually mean? What do we mean low concentration and high concentration? In the diagram, which, the left side or the right side, which one has a higher water concentration? Well, water concentration simply refers to how much water, how many water molecules there are in a given volume. And so the question that we could ask, which has greater water concentration, fresh water or salt water? Now, by fresh water, I mean maybe distilled water, 100% water. So if I have a glass or a beaker of 100 milliliters of distilled water, it's 100% water molecules, and I have a beaker of salt water that is, let's say, 98% water molecules and 2% salt molecules, which one has a greater concentration of water? Well, the distilled water one does, the one that has 100% concentrate or 100% water molecules. All right, so now let's talk about what happens when we tie this into cells and maintaining homeostasis. What happens when cells get put into environments that are not the correct environment for them? So we'll start with the one that is the correct environment. Isotonic. The prefix iso means same. And basically an isotonic solution is when the concentration of things outside the cell are the same as concentration of things inside. Or you could say when the concentration of water is the same outside as is inside. If you look at the diagram down at the bottom, the isotonic solution is the one that's in the middle. If you see the arrow, there's an arrow going into the cell and there's an arrow coming out of the cell. That indicates that water is coming and going at an equal rate, and we call that equilibrium. The red blood cell in this diagram is staying the same size. That's homeostasis, staying the same, and that's what cells want. So what happens if cells get put into a different solution? We'll start with the first one called a hypotonic solution. This would be a real classic example is if you were to drop a red blood cell into distilled water. Distilled water, again, 100% water molecules. So this is where the water concentration is lower in the cell, so water rushes in, causing the cell to swell. Imagine for a second in the diagram where it says hypotonic, imagine that the water that it's been placed in is 100% water. And imagine that the amount of water in the cell initially is 98%. So the water is going to go from high concentration, where it was on 100% outside of the cell, to low concentration, where it's 98% inside the cell. That causes water to rush in, and the cell starts to get bigger and bigger and bigger. A student helped me to remember the word hypotonic. This was like 15 years ago. They said, well, KJ, when I was trying to learn hypotonic, I remembered the word hippo. Hippo. When I think of a hippo, the animal, a hippo, I think of a big, huge, bloated animal. And that's what red blood cells look like if you put them in a hypotonic solution. Well, I thought that was brilliant, and I've been teaching students hippo ever since. So if a cell, an animal cell, gets placed into a hypotonic solution, the cell gets big, 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 and eventually the cell bursts. That's deadly for cells. They need an isotonic solution. In the opposite, this is what would happen. A hypertonic solution is what would happen if you got stranded on a boat in the middle of the ocean and all you had to drink, keep in mind, all you had to drink was the salt water. There's no pop, there's no fresh water, there's no coffee or tea, just salt water. Salt water has a much lower concentration of water than what's already inside your cells, which is a really weird concept, but just trust me, it's true. So look at the third diagram. If you place a red blood cell into salt water, there's less water on the outside. Let's say the number is 95% water on the outside of the cell, and on the inside of the cell, it's 98%. Water travels from high concentration to low concentration. So the water would escape out of those cells. It would cause the cells to shrivel. And that's what happens when a person drinks only salt water. Gargling salt water, no big deal. Drinking a little salt water, no big deal, as long as you have fresh water that you can use to rehydrate yourself. It's when you only have salt water that it becomes a problem. All right, now this picture here is just a summary real quick that shows what we were just talking about. I'm going to skim ahead. Now here's where it changes a little bit. Remember that human cells are soft. All they have, or animal cells are soft. All they have is a cell membrane on the outside. But plant cells have that cell wall. So that makes them extra tough. So if we put, and if you look at the diagram or look at the photographs, if we put a plant cell into a hypotonic solution, like distilled water, water's going to rush into those cells like they did in the first picture on the left. And the water fills up the cell, and the cell membrane is pressed against the cell wall. But the cell never pops because it's got that rigid cell wall. In fact, the cell can't even get any bigger because that cell wall keeps it the same size. So what it does is it increases the pressure inside the cell. We call that Turgor pressure when it's in a plant cell, and that's actually good to the cell. You know what? I'm going to have to stop this in about 30 seconds, so I got to talk fast. It's good for the plant. Hypertonic solutions, which is what happens on the right, is when we put plant cells in salt water and it sucks the water out. That's what happens in Minnesota in the winter. We put salt on our roads and the water causes the plants to shrivel up. That's bad for plants. All right, good luck. Everybody have a great night.